Hi, my name is Hannah Zhao, and today I'm very excited to talk to you about my project, which is on investigating sulforaphane isocraniac on hindering non-small cell lung cancers and IF2 mechanisms. In the United States, a projected 23,000 people will be diagnosed with lung cancer by the year 2025. In the year 2021, there's an estimate of new diagnosis of lung cancer every 2.2 minutes. Out of all types of lung cancer, non-small cell lung cancer is the most common type, which accounts to 80 to 85% of all types of lung cancer is non-small cell lung cancer. And figure one is shown, there's different types of non-small cell lung cancers. So for example, non-small cell large cell, non-small cell lung cancer, and non-small cell acroneus. My study mostly focused on non-small cell lung cancer, which is used and represented by the cell line SW1573. Sulforaphane is an anti-cancer activity which is mostly attributed to isocraniac vegetables such as broccoli, cabbage, and kale. Sulforaphane enters the cell membrane, which binds with KEP1, interacts with NRF2 in the cytoplasm. Through sulforaphane binding with KEP1, this promotes the mitogen activated protein kinases, also known as MAPK, and the protein kinase C, which is promoted by the interaction of sulforaphane to KEP1, as I said before. This promotion leads to the activation of NRF2, which is a competitive inhibitor of EGFR receptor. This promotes, once NRF2 enters the nucleus, this promotes MAF, which is a transcription factor, and this regulates the transcription of cytoprotective genes. The anti-cancer activity, as shown before, is mainly attributed to cruciferous vegetables. Morphine is a novel inhibitor of epigenetic growth factor receptor expression and is effectively inhibiting cancer growth. And previous studies have shown that it is mostly inhibited in cancers such as prostate, breast, and oval cancers. EGFR is known to be highly expressed or mutated in roughly 60% of non-small cell lung cancer patients. This has EGFR is mutated in my grandmother who got non-small cell lung cancer in the end of 2018. And NRF2 is activated with simulation of EGFR. This leads to my purpose. First, I want to determine sulforaphane, which is a chemoprotective chemical, can inhibit specifically lung cancer, specifically non-small cell lung cancer. Number two is to identify how sulforaphane binding with NRF2 can inhibit non-small cell lung cancer growth. Number three is the proliferation of sulforaphane when sulforaphane introduced to non-small cell lung cancer cell line. The cell line that I used in my study is SW1573. So first, I did MTT assay, also known as the proliferation assay. This tests the cell metabolic activity of non-small cell lung cancer after 24 hours introduced with sulforaphane, which later on employs to measure the chemicals effect and metabolic activity and viability of cells. As shown in figure 3, the high concentration of sulforaphane, which is introduced to non-small cell lung cancer, the more percentage of cell death, with a p-value less than 0.05, which is supported with 1 star, and 0, which is less than 0.01, so it's more than 2 stars, versus the control group. Sulforaphane is reduced cell activity, which on average is by 34.5% in non-small cell lung cancer. Values are suppressed and tested with a standard n equals 3. The assay that I did is the migration assay. As shown in figure 4, the migration assay is used with 3 horizontal lines that simulates the wound and placed under a microscope at 40 magnification was observed. And after taking pictures, ImageJ was used to gather the final results. As shown in figure 4, the more percent of sulforaphane, dilutions of sulforaphane added to non-small cell lung cancer cell line, the less cell of proliferation, shown with the picture above. With more addition of the sulforaphane, especially in picture number 5, above sulforaphane with the dilution of 10, which is at the highest concentration, the gap between the two cell membrane is more apart. Migration cell assayi counts how many cells pass through the porous membrane. And the third assay that I did is a molecular docking assay. By using PyRX was used to determine the affinity binding size of the micromolecule and sulforaphane. I tested all different types of NRF2 shown in table 1, and the highest concentration, the highest affinity binding that I found was that oxidortase, which is 401770, a type of NRF2, has a high affinity binding, which is at negative 7.3. And this affinity binding shown in figure 5 is right here and enlarged right here. The purpose of the docking is to show the most bounding mechanism and whether or not sulforaphane can inhibit the likelihood of more growth to non-small cell lung cancer on a molecular level. And finally, the assay that I did is caspase-3 assay. And this assay, as shown in figure 6, at a lower concentration, it shows that sulforaphane creates downregulation, but at a higher concentration, sulforaphane has shown that it has a 90.225% of apoptosis with non-small cell lung cancer. And the caspase-3 assay measures the effect of sulforaphane apoptosis behavior on non-small cell lung cancer. And this is tested with a standard deviation at n equals 3 and a p-value of 0.05. In conclusion, oxidortase with the shown of sulforaphane, KEP1 and NRF2 is inhibited. Inhibits EGFR, and this can create promotion of earlier protective activity to promote. Sulforaphane reduced 34.5% of cell viability and proliferation in non-small cell lung cancer cells. Oxidortase, which is very highly expressed under stress conditions, is deoxifies chemical stress in normal tissue, which provides cytopenia.